My name's James Chiguli Mukasa, the acting head of this department of microbiology, Soroti University. Where we are now, this is the microbiology preparation and research lab. We have a number of equipment in this lab. We have a number of reagents and chemicals, other supplies, which enable us to perform the teaching. The equipment you will see around here are basically meant for teaching and carrying out research. We have a number of equipment, including incubators, we have refrigerators, we have -80 ultra freezers for the storage of microorganisms at very low temperatures. We also have ovens, we have microscopes, we have weighing balances, we have orbital shakers. We have the biological safety cabinet level 2, which is a requirement for containment of microorganisms that must be handled at level 2. So we are actually protected here. We have enough glassware, we have enough materials to safeguard ourselves, including we have gloves, we have face goggles, and so forth. And for safety, as we shall see when we move to the other side, we are equipped with the eye washing center and the first aid kit. Inside here we also have fire extinguishers. In case of fire, we have the appropriate classes of fire extinguishers that would help us in case there is a fire hazard. Yeah, where we are now, this is the Microbiology Teaching Lab. As you can see, we have benches. We have 2 benches in the middle. We call these island benches because they are isolated. Then on the wall, we have a whole bench the other side and a whole bench the other side. Now on these benches, we have services like water, we have gas and electricity. So the students are able to access all those services. So as you can see, we have organized microscopes here and also some staining centers where they are supposed to be. Because of the COVID pandemic, we bring in very few students. Now these are 25 microscopes, so we bring in 25 students at a time so that they don't congest, and we ensure that actually they have masks and they are all protected. We have the sanitizers and some other antiseptics and disinfectants. So we organize them such that they work in groups. You realize that at a particular group, there is a microscope, Bunsen burner, a staining rack, there is a timer, a inoculating needle. There is also a set of reagents that you are supposed to use for staining.
My name is Nkanjuanga Eliasaf. I'm employed here as a Chief Laboratory Technician, Department of Microbiology. So, our main role, the university is still young, and for the Department of Microbiology, we started our practicals this semester. The microbiology course starts in 2nd year. So far we have done 4 practicals. We use it for shaking samples. For example, when we are doing the test of syphilis, VDRL, we test for typhoid, widow. Put your sample here. Then mix it, the sample and the reagent, then you put it here and shake it for the prescribed time. 2 minutes, 5 minutes, 8 minutes. It rotates like that until the time is over. Then when it is over, you switch off. Under conditions where there is no oxygen, almost. That's why they're called anaerobic. And after putting our organisms on those plates, we put them here. It has got a lifting rack. After cultivating the organisms in those media, you load them here. If there are more, you put more. Then you put them here. Then get this gas generating kit, which contains a carbonate and acid in powder form. You cut here with a scissor. You add, sorry.
No, just elaborate.
You cut here with a scissor. Then you add tap water as recommended by the manufacturer. You add the volume of water which is required. Then you put it here. Then put it inside. When you have already added the water, carbon dioxide will be generated. Cover, then put the whole thing in the incubator. Then there you will be